Okay. Hello. Let me go back. All right, I'm ready. I'll start now. I'm Mia Curic. This is my HuBMAP project. I work for the Jane Lab at Washington University, and I'm doing my project on light sheet fluorescence microscopy and its uses in mapping structures of the kidney. A little bit about me. I'm from St. Louis. I go to St. Louis University, and I'm going to be a sophomore this next year. I'm a biochem major on the pre-med track. I haven't had really much research background, so I thought that this program would be wonderful for me to gain, more knowledge on the scientific process and whatnot. Main goals of the lab. So I'm working for Dr. Jane's lab at Washington University in the nephrology department. He's working with HuBMAP and other organizations and programs to investigate the kidney structures and functions. I know that HuBMAP, we're more focused on creating the spatial atlas of a healthy human body at single cell resolution. And he has many different experiments going on, including single cell nuclei isolation analysis. But I was more focused on in my project, imaging and mapping of larger scale tissue samples and sections. And through immunohistochemistry and performing immunofluorescence, I was able to use light sheet fluorescence microscopy and confocal microscopy to create high resolution 3D images, and which led me then into image analysis and whatnot. So some objectives for my project. I wanted to ask the question how the different structures in the kidney were organized, as well as how these structures were visualized and then interpreted into data. So just a little bit of background with the wet lab, because I was involved in wet lab. This is just sort of like our basic protocol for wet immunostaining and whatnot. You would prepare your sample, whether it be formalin fixed paraffin embedded tissue or frozen tissue. You'd retrieve your antigen, you block your background or any non-specific, making sure non-specific antigen binding doesn't occur with antibodies. Then you would detect your target, which is just basically antibody labeling, as shown in this diagram here. Like you have your primary and your secondary, and you have a fluorophore on the end of your secondary, which would help you to visualize your sample. And that led me into my image analysis. Some specific antibodies that I use, I just wanted to go over maybe two. I was more looking at neurons and glomeruli in the kidney, or in the nephron, I guess. The glomeruli is really important for, filtration and whatnot inside of the kidney. So I used NPHS1, which is just nephrin protein, and it's an antibody that stains for the podocytes that are surrounding the glomeruli inside of the Bowman's capsule. Or podocalyxin was something we also used to help stain. That also stains for vasculature. And another main one, a big one for the neurons, we used TUJ1 and TH, which were just class 3 beta tubulin protein antibodies. So that would help us stain for the neurons. And there's many others, including AQP2, which is an anti-aquaporin antibody, and that help you stain for collecting duct and whatnot. Anyways. This is just sort of an example of a kidney dissection that they would perform. And this is an example of one of, a tissue sample that we would use then for light sheet fluorescence microscopy. And then this is an example of, this one was my sample of acute kidney injury before and after tissue clearing, which is just preparing. This is while you're preparing the sample to be, stained and image. So this is before imaging occurs. You'd have to stain it, but you want it to be clear before you stain. And then this is more of what my project was about. I started with imaging, kind of figuring out what kinds of imaging were best and would work best for me. We looked at traditional fluorescence microscopy, and you'd have light coming from both the focal and plane and out of focus areas. Like this is conventional wide field microscopy. This leads to a lot of background fluorescence and can cause unreliable imaging, sample damage, and poor image quality. And so all alternatives would be confocal microscopy and light sheet fluorescence microscopy, which is what I'm going to be focusing on more. Just a little background on confocal. It uses pinholes to block out of focus light. and it detects light from the focal plane, which we can see in 2A. Out of focus light gets blocked by the pinhole, but it's not detected, which can lead to the sample getting damaged. So you see that. It handles samples that are only a few hundred micrometers thick, and it takes very long imaging 3D. So why light sheet fluorescent microscopy? It uses optical 2D slices to create high resolution 3D images. It utilizes a technology called single plane illumination microscopy, and it works with laser beams perpendicular to the imaging system. Like in figure 1D, we can see this is the light sheet perpendicular, and then this one is confocal. You can see the pinhole, and then this is conventional wide field microscopy. Its excitation light is in the form of an hourglass. This kind of leads to less out of focus light in comparison to the conventional and confocal microscopy. And it can also scan tissue several centimeters thick. So that's, an advantage, as well as scanning probably twice as quick as confocal for the 3D imaging. Figure 2, you can see the light paths for the confocal and the light sheet. And then this is just sort of a flow of what I did in my image analysis. There's many other, there's tons of different software out there, but I used Stitchy to stitch images just, after, I don't know if you guys can see this. Okay, I'm so sorry. And then you would move on to, after you've stitched your images, to, create a nice image after you've, used the microscope. You would have to segment out structures that you wanted to be kind of amplified, or what signal you wanted to be amplified in your image. Whether it be nerves or, different structures. So I use image J for more filament-like structures, including nerves. It could also be used for collecting ducts, proximal tubules, and whatnot. This was sort of my process in that, how I segmented it, and then how I viewed and binarized the images. I used Python for segmenting out the glomeruli structures. And then after all this, I would use Maris to and more interactive software. It cleaned up and visualized it pretty well. And then leading on to, in the end, I didn't really work on creating a network, but that probably is something else I'd like to do in the future through Python. You can just kind of create the next connections and show connections between different structures, which is really neat. Let's see what's next. What I did, these are just my questions, so you guys can look at them again, but I interpreted different structures from the light sheet fluorescence microscopy, and I used different programs to quantify and interpret these visuals into data. I used different software tools and algorithms to segment these structures and make them pop in my image, as well as I used quantitative analysis. I did quantitative analysis of the human kidney. I'll show you in a little bit about my morphological properties of glomeruli, for example, volume that I found from these softwares. And then also through a networking, you're able to create communities and connections. You're able to showcase those connections between different structures in your image. So this is more my project. You can see right here, this is my glomeruli segmented out. I made them really nice, cleaned them up, and I also had them with the nerves here. And then this was with a network. This was my sample was a acute kidney injury sample also, just to note. I have an animation that I think I'll get to at the very end to show you guys. So this is just sort of, I was just wanting to pull up, I was trying to show you what all, you could do a lot of things through, this was through Amaris, but you can do so much through these softwares, finding morphological statistics for the glomeruli in my tissue. I was able to make graphs of area, volume, sphericity, and then even another project could be comparing it with some other sample. It could be like this one's chronic kidney disease. You could also compare it with healthy tissue. Some conclusions I'd have to say, through the light sheet fluorescence microscopy, one can create high resolution 3D imaging, and this helps to identify structures within tissue, not only kidney, but all sorts of tissue. With the use of many software programs, one can quantify and interpret these visuals into data. I think it's extremely helpful for not only learning the structures, but their functions and how they develop throughout the tissue. This can lead to different projects about these structures' connections, their growth and development, or aging over time. So I think it's definitely important. I had a lot of challenges learning different softwares for imaging and analysis, being comfortable in a wet lab setting. I had to fail a lot to learn. But then I also learned that you really need to speak up when you don't know something. So yeah, and then some acknowledgements. Thank you all for joining this call and whatnot. I also have to thank my lab mentors and PI. They're really great with helping me out with everything. So yeah, my resources. You guys have any questions? And I can't even see myself.
Are you going to show a movie?
Oh, my movie. Sorry. Thank you. Thank you, Dr. J. Hold on. I don't know how to get out of this. Pause, share, and show. Okay. Okay. Hold on. Where am I? Oh, okay. I pulled it up here. Okay. This is my animation. Thank you for reminding me. All right. So this is my glomeruli. Hello?
I think it maybe oh, was there any audio on the video?
I don't think so. No. Oh, okay.